this is where we left off. All right, life cycle, monocystis lumbrica. All right, a couple of housekeeping things. We got lab. If you give your presentation, make sure you get it to me before lab so I can download it and have it posted. We'll have a quiz in lab. And then next week we have a practical. Next week we'll also have our exam, our third exam. And then I think it'll be good because then our final exam will be the week after that. So kind of rapid fire at that point. I have 2 more quizzes posted. The other flagellates, that should have been posted on Friday. I thought that was already posted. I was surprised it wasn't. But I posted that one. That's I think 10 minute time limit, 6 or 7 questions. That's due at the end of the day on Wednesday. And then I also have the amoeba. That was posted. That's like 5 minutes. I think there's 4 questions. Because the amoeba was just focused on the life cycle, really event amoeba, and we see it in the lab for some of the structures. And then we'll finish this off. In lab, we will look at monocystis lumbricae. We'll dissect it out. Dissect seminal vesicles, real easy to see. You'll see working with freshly killed organisms a whole lot better than preserved organisms. So check those out and try to find the different life cycle stages. Now in the life cycle stages, these are the Gregarines. We do have some additional terms. So our oocysts are going to contain our sporocysts and sporozoites. All right. For us, oocysts could also be considered the spores. So they're going to develop inside our gametocyst. And you're going to see naming conventions of how these things operate a little bit different as we move forward. All right, so did you have any questions? Oh, will we have a quiz in lab today? Yes, we will. So good review for the, it'll be a good review for the practical. All right, any questions on monocystis? You'll see it, plenty of pictures online. All right, we're going to talk about Gregarina. With Gregarina, we do have a slide of, I think it's just labeled Gregarina. So what we're going to do is focus on Gregarina cuneata. This is an intestine of mealworms, so Tenebrio molitor. This is similar for a lot of other gregarian parasites, so other Gregarinas. I just picked cuneata because we can kind of define a host as a mealworm. All right, we could probably purchase these from Carolina Supply, and they probably would have these Gregarine parasites. Haven't ever really looked at them. Now, what's interesting about the Gregarina is that we're dealing with septate Gregarines. So when we look back here, our 2 suborders, we've got A. septitorrhina and septitorrhina. So this is our septitorrhina, which means we're going to have 2 different body plans here. We're going to have one body up here, second body down here. Actually, here's our body. Our individual body is in 2 parts, and here's our septum. That's our septate Gregarine. This is actually in our 2 gametes that are fused in syzygy. So in our body, we have broken into 2 parts. We've got a protomerite, and the posterior part is the deutomerite. And we've defined them as anterior and posterior because these emerge from the internal cell. And there's going to be one other attachment called the epimerite, or I've heard some people call it epimerite. But that kind of remains embedded, and then when these things fuse and they detach, the epimerite ends up detaching from it. So I have an epimerite down here. All right, so these are the gametes. With our life cycle stages, when they fuse, our gametes will fuse in this format where we have the anterior gamete, that's going to be called our primate, and our posterior gamete, which will be the satellite. We can see these structures on our slide. And these will ultimately fold in upon themselves, secrete a cyst wall, and that forms our gametocyst. All right, and then we'll undergo gametogeny, where these do have two different mating types. So they will produce, one is basically the macrogamete, or the macrogamete, and then the other one will produce all these microgametocytes. And then you'll get your fusion and your production of all of these oocysts inside of the gametocyst. And while we're at it, in a lot of these guys, when you get the gametocyst gets secreted, it undergoes a process of dehiscence, called dehiscence, where it extrudes these oocysts in like a string-like fashion. Think like a pearl necklace, like a string of pearls. These are all the oocysts that are coming out. Now, they could be coming out individually, or they could be coming out in like a tube-like structure. But this is the extrusion of oocysts is called dehiscence. So I know we could probably go out when it warms up, catch some, let's say, dragonflies, some damselflies, put them into a test tube, and in like 24 hours, some of these gametocysts may actually appear on the slide. Then you have to put them out and get them into a proper humidity type environment. Oops. Oh, can't see the slides. I wonder. I have everything. Here's the slide. Sorry about that. Had it up here sharing. All right, here's the slides. I guess we'll have to go back. Here's our gamete. There's our septum. The gamete is divided into a protomerite and a deutomerite. And then the epimerite, or epimerite, would be at the very anterior. That remains embedded in the cell. That gets detached when these things detach from the gut epithelium. This structure is our two gametes that have fused. That syzygy is a fusion. All right. The anterior one is our primate. Posterior one is the satellite. And this is the one. We'll see these on our slides. And then, pointing this out, this is our gametocyst. And through a process called dehiscence, you'll extrude all of the oocysts out in these long chains. Or it'll be a tube that has an oocyst in those tubes as a long chain. All right. Pick that up. So, I'll leave that up. We're going to go over the life cycle. We're not going to see any of them that have an epimerite on them. So we can just kind of imagine what it looks like. We're basically just going to see these gametes on our slides. Okay. Let's enlarge this. Hide the slide again. All right, Gregarina cuneata. We're going to have our oocyst. All right, that's our mature spore. Our mature spore, our oocyst. In Gregarina cuneata, it's actually kind of box-like. Kind of what our oocyst would look like. All right, so the mature spore is out in the environment. It gets consumed by our host. So we have our oocyst in the gut right now. All right, so the oocyst is going to contain our sporozoites. So inside our gut, our oocyst is going to get released, or our oocyst is going to rupture, releasing the sporozoites, or you can say sporozoites escape. And these sporozoites are going to attach to the gut epithelium and penetrate. Right, attach and penetrate the gut epithelium. Sporozoites will then transform, or these sporozoites will transform into a trophozoite, which is our feeding stage. All right, and in this feeding stage, we will start to have growth of the organism. The sporozoite penetrated it, transforms to our trophozoite. Trophozoite's now going to be feeding, absorbing nutrients, enlarging in size, and this is both intracellular and extracellular growth. All right, so you're going to start having part of this trophozoite protruding from the gut epithelium. All right, and then as it's developing, as it's exhibiting this extracellular growth, you're going to start to have differentiation. All right, you're going to have differentiation into our gametes. All right, gamete. All right, we're not going to know what gamete type it is, if it's going to be an anterior one or a posterior one, but they've differentiated and they are kind of fixed at that point. One is already now determined to be the primate, one is determined to be the satellite. All right, now these things are attached to the gut epithelium. All right, and they're being attached because they have the epimerite embedded into that cell. They're going to then fuse, or at least come up to each other and fuse and then reorient. So one of them will kind of contact, detach, and then fuse into that structure. So this is syzygy. All right, now we have our paired gametes. All right, still attached. All right, still attached. Then they will detach and lay down a cyst wall. Detach and a cyst wall. All right, so at this part with our paired gametes, we're going to get that classic primate-satellite formation in the lumen of the gut. It adds the cyst wall, and what this will do is basically fold in upon itself like that, so that it deposits the cyst wall. Now we have our gametocyst. And our gametocyst has those gametes. Make sure we're on there. Yep. Gametocyst has our gametes. Now, once we have our gametocyst that has the gametes, now we undergo gametogamy, all right, to produce our macro and our micro gametes. So gametogamy, we've got our macro gametes, micro gametes, and note that we are in the gametocyst. All right, so We are still inside of our gametocyst when we have gametogeny producing our macrogametes and our microgametes. All right? These things will now fuse to produce our zygote. Gametocyst with the zygotes. Now, our zygotes are going to deposit a cyst wall. So now we have gametocyst, our gametocyst, with oocyst. So the zygote secreted the cyst wall. Now we've formed the oocyst once we have that cyst wall. All right? And then once we have this, then we can have our sporulation followed by sporogony. All right, sporulation followed by sporogony. So we have our gametocysts with our sporulated oocysts. And this stage gets released in the feces. All right? That gets released in the feces. So our host here is our mealworm. We go out into the environment. Now, we still have our gametocyst with our sporulated oocysts. So our oocysts have those sporozoites. And so gametocyst with the oocyst with our sporozoites. Then we have dehiscence. That now releases our mature spores, which is the oocyst with our sporozoites. With our sporozoites. All right. Questions? Plenty of Gregarine out there. Definitely. See if we have any questions up here. We good? Right. Pathology. Little is known. It doesn't infect humans. A lot of people can care less, unfortunately. Could take, speculate as to possible effects on the host. Could have intestinal inflammation in the insect. You have sporozoites invading the cells, right? They're growing out of the cells. When they detach, it's a possibility that you're leaving damage on that gut epithelium. Damage to gut epithelial tissue, you could influence absorption. So perhaps this is the cost. Perhaps these individuals that are infected are less able to utilize the energy that they consume, or less able to assimilate the energy that they've taken in. Just speculation. We've been pretty consistent on talking about pathology, so I thought I'd include it here. All right. Any questions? Nope. All All right. That wraps up the Gregarines. Next up is Coccidians. Oh, I messed that up. Didn't change that. That's better. That's better. Coccidians. So with our Apicomplexans, we talked about class, Conoidacida, right, and the Aconoidacida. If you notice that in the Conoidacida, we had another order that we were going to talk about, Eucoccidiorida. Eucoccidiorida. These are the coccidian parasites. Right? And the ones that we're talking about are all members of the subclass Coccidiacina. Coccidian parasites. It was in the one of them was in the news. I was holding off until we had our presentation, but it hit the news. Read about it today. All right, so these are small intracellular parasites. They're going to live in a variety of cells, and they're going to infect invertebrate and vertebrate hosts. Let's be clear, though, that individual species isn't going to infect invertebrate and vertebrate hosts. It's that there are different species, and the range of species that we have include parasites of invertebrates and parasites of vertebrates. All right. So in this group, we have three major phases of replication: merogeny, gametogeny, and sporogeny. Right, so merogeny is the production of merozoites, and gametogeny is the production of gametes, sporozoites, the production of sporozoites, or sporogeny is the production of sporozoites. Just kind of as a quick review, so what we'll see. Now, a lot of this is multiple fission. However, we're going to introduce something a little bit different. Something a little bit different as well. Ready? All right, so our general life cycle pattern. This is general. We're going to diagram some life cycles. The general pattern is that our sporozoites are going to invade cells and become trophozoites. Our sporozoites are going to be rod-shaped or banana-shaped. Right. Trophozoites are going to feed on the cells, they're going to enlarge, and then they're going to undergo merogeny to produce the merozoites. The merozoites are going to escape the cell and then enter new cells. This escape is traditionally rupture of the cells, destruction of the cell. So we've got a site where the release of these merozoites could produce pathology. All right, and then when these merozoites enter the new cell, they're going to transform back into trophozoites, feed a little bit, get larger, and then undergo merogeny again, and it's just going to be a regular cycle. It's going to be a regular cycle. Ready? Yep, almost. And again, I'm just providing the general pattern that you can understand. These next life cycles are probably the most complex that we have. All right. So we have this cycle: merozoites reinfecting cells and undergoing merogeny. Right. Some of these merozoites, after they infect the cell, will undergo gametogeny. All right? And these things are defined. So some of these merozoites will undergo gametogeny to produce microgametocytes. The microgametocytes are basically your microgametes. The microgametocytes are going to produce our microgametes because they'll undergo multiple fission as part of this gametogeny to produce a bunch of these microgametocytes. Many of them are biflagellated, right? So they've got two flagella. And then these microgametes are going to rupture the cell to get out. Now, most of our merozoites end up being these microgametocytes, transforming to the microgametocytes, undergo multiple fission and produces these microgametes. The rest, after the merozoite infests the cell, will transform to a macrogametocyte. These macrogametocytes will basically undergo gametogeny to produce the macrogamete that is a single large ovoid body with a central nucleus. So if you think about it, our merozoites, if they become a microgametocyte, they're going to produce a whole lot of microgametes. But if a merozoite becomes a macrogametocyte, it's one. It's one macrogamete. All right? Fertilization is going to happen. So our microgametocytes are then going to penetrate into the cells that have the macrogamete. That's our fertilization to produce their zygote. All right? And then once we have our zygote, in a lot of cases, not always, but in some of the cases, we're going to lay down a cyst wall and then undergo sporulation and sporogony. So we're undergoing the zygotic meiosis again. That's reducing our chromosome, or our genetic material in half. It's reducing our chromosome number back to the haploid state. It said that it could possibly be in a sporocyst, inside of an oocyst. But it doesn't always have to be. It's like Plasmodium. Malaria doesn't have that oocyst stage. Doesn't have that cell wall. I mean, it does have the oocyst stage. It doesn't have the cell wall that's characteristic of it. So this is the general pattern. And I encourage you, once we start getting our life cycles up here, go back to your notes for the generalized life cycle pattern and mark, note on those life cycles where that pattern, where we see that pattern. I think it'll help you learn these. Ready? All right, so we've got two coccidian parasites to discuss. One is Eimeria. We don't have any specimens here. We talk about it because occasionally we have some of the ag students come in, take the class. This is very important for chicken farmers, for poultry farmers. All right. The other one is Toxoplasma gondii. So we're going to start with Eimeria. I've got a diagram of Eimeria. This is our oocyst. And as I said on the previous slide, you can have a sporocyst inside of the oocyst. Yep, here's our oocyst. These are sporocysts with sporozoites inside of them. All right. Characteristic, the shape, how many sporocysts, how many sporozoites, do they have these residual bodies inside sporocysts? Are they inside the oocyst? Do they have polar granules? All this stuff helps with identification. All right, so in Eimeria, these oocysts develop very resistant walls. Right? These are very resistant to disinfecting conditions. All right, which means if you get it in a hen house, it's going to be hard to get rid of. All right? And especially if you get it in a hen house, the pathology itself, you'll know it. I mean, good chance that you're going to lose that entire coop. All right, this oocyst wall is resistant because it has two layers. The layers consist mostly of lipids and proteins, so it helps block out both the polar and the non-polar disinfectant products. All right, they're also resistant to proteolytic enzymes, so you can't really even go in with some of those enzyme treatments. All right, they're really resistant. Now, this is just Eimeria species. All right, there's multiple species of Eimeria that are out there. Multiple species infect chickens too, as well. Now, many of these things have a tiny opening in the oocyst. That opening is called the micropyle, and then it's capped by a micropyle cap. All right. Trying to can't really see any in our image. It would be at the anterior end. There's maybe one. You can almost see the opening. But they've got this micropyle cap. It helps with identification. All right. As I said, many of these species form a spore cyst inside of the oocyst. All right. And in the spore cyst, instead of having a micropyle or a micropyle cap, their cap is called the stata body. Host enzymes, stomach enzymes and small intestine enzymes will dissolve the stata body. That allows our sporozoites to get out. All right, so when you get into the proper host, it'll dissolve it, and that's how our sporozoites can get out. Some of them also have these residual structures. There's an oocyst residuum, a sporocyst residuum. This is basically leftover cytoplasmic material. Hasn't been incorporated in any of the sporozoites or in the oocysts, sporocyst or the sporozoites. All right, this is mostly lipids. It's thought that maybe these things could serve as like an energy supply when the spore leaves the host. But this is also useful in identification. When these oocysts get out of the host, typically that is when sporulation will occur, and sporulation and sporogony. So a lot of the times that this process of sporulation to produce our sporocysts and sporozoites is oxygen dependent. So they need to get out of the host before that can happen. All right, we don't have any of these slides, unfortunately. But if you go to like vet school, or try to go to be a vet tech, you'll probably see some of these. Ready? All right, so Eimeria is usually host name location specific. All right, so one species of Eimeria typically infects one species of host, and they are very specific on where they infect. So here's a chicken diagram, all right, just to kind of show you where each of these different Eimeria species infect. So like Eimeria hagani is in the duodenum. Eimeria necatrix is in the small intestine, large intestine. Eimeria burnetti. We're going to talk about Eimeria tenella because that infects our cecum. All right, and that's the one that we have a life cycle for, or that we'll go over the life cycle. These species are going to vary in their pathogenicity. Some of them are really bad, some of them not so much. But some of these, are notable because the infections tend to be self-limiting. So it's almost like there's pre-programmed number of replication steps that will take place. And as you'll see with Eimeria tenella, if the chicken can survive to the point where they start shedding oocysts, then they're typically going to survive the infection. All right, if you think about it, that means then that this is going to cause pretty significant death. Do they infect humans? No, not the ones we're talking about. All right. They're just important because they affect our ranching farming operations. As I said, you have a chicken coop with 10,000 chickens and you bring one or two that has Eimeria in it, you're basically going to have to lose. You're going to lose all those chickens If Eimeria doesn't take it out, then you're going to be required to kill it to prevent spreading from other places. And you're going to have to go in and really disinfect. Flame disinfect and infecting. Pretty common. All right, so one we're going to talk about is Eimeria tenella. This is in the epithelial cells of the intestinal ceca of chickens. It has the potential to infect the entire coop, often causes death in our young chickens. There's no treatment, only prophylaxis. That's why in the young chicken feed, they include a coccidiostat, which is an anti-coccidial drug. It tries to kill the parasite before it establishes an infection. They do have some vaccines being developed, but they show mixed results. All right. Major downfall to this is the expense related to producing the vaccine, so mass production of the vaccine, and then also administering the vaccine, because you basically have to catch every single chicken and vaccinate it. It's a huge manpower, huge expense to do that. That's why the coccidiostats in the young commercial feed tend to be the most common thing. It's a lot cheaper to do this. The other downside of vaccines is that the effectiveness based on the strain and species of Eimeria. So in Eimeria tenella, which we'll talk about, there's different strains of that. So some strains may be more effective, a vaccine may be more effective against certain strains of that Eimeria tenella, but not so much against others. Also, kind of depends on the breed of chicken. Do you have broilers? Are you doing egg layers? What specific breed of chicken do you have? So all these things, again, a lot of people like to talk about vaccines as a holy grail. Parasites have been around for a long time. They've done well at evading host's immune system. It's a little bit naive to think that we can just conquer parasites with a simple vaccine. So it's difficult. All right, let's go over the life cycle. Life cycle is probably going to be one of the most complicated. Relatively simple in terms of directions, but it is self-limiting. And I hope to be able to get it all on this board. All right, so this is Eimeria tenella. Eimeria tenella. All right. We're going to have our sporulated oocyst. That's going to be in our environment. All right. This sporulated oocyst is an oocyst, a double wall oocyst, that's going to have four sporocysts, each with 2 sporozoids. Again, probably don't need to have it. 4 sporocysts, 2 sporozoids. All right. So our chicken is going to ingest this sporulated oocyst. All right. The host digestive enzymes are going to dissolve that state of body, releasing our sporozoids. Sporozoids. It's in the lumen of the gut. This will then penetrate the gut epithelium, penetrates the gut epithelium to the gut mucosa, the lamina propria. So the gut mucosa is just collectively the entire tissue of the gut, right? Not the outer epithelial layer. Well, it includes the outer epithelial layer, but we're going a little bit below that to the lamina propria layer. All right. In here, our sporozoite gets phagocytized. All right, it gets phagocytized by a macrophage. All right, so now our sporozoite's inside a macrophage in the gut mucosa, and this macrophage will transport our parasite, that sporozoite, to the crypts of Lieberkühn. And again, we've seen this before, right? Crypts of Lieberkühn. All right. So we transport to the crypts of Lieberkühn. We still have our sporozoite in the macrophage. And that sporozoite will now escape and penetrate a new cell at our crypts of Lieberkühn. So now we have our sporozoite in the cells of the glands of Lieberkühn. Glandular cells. That's where we're at. Glandular cells of the crypts. Once we get in here, now we transform to a trophozoite. All right. Transforms to trophozoites, feeds, enlarges, undergoes morphology to reproduce our merozoites. And again, still in our glandular cells, we produce our merozoites, and then we will rupture. So release the merozoites and reinfect. Reinfect the cells. We are at the site where we need to be in the gland, in the crypts of Lieberkühn. We're there now. So the merozoites, okay, trophozoites underwent morphology, produced the merozoites. These ruptured the cell, so they caused them to release. All right, and how many of these? About 900. That's how many we get. So they're going to be really small. So then we're going to get in our release and reinfect, all right, we're going to get our second generation. Infecting the cells. So, first generation merozoites, they infect, they produce via morphology our second generation cells. These will also release and reinfect. However, these are only about 200 to 300. So they're a little bit bigger. So you're not getting as many. All right, and then this is going to give us our third generation of merozoites. All right, so they're going to penetrate, again, release and reinfect. They're going to undergo morphology to produce our third generation of merozoites, which then cause our cells to release and reinfect. All right. Release and infect, and then undergo gametogamy. So now we get our microgametocytes and our macrogametocytes. All right. Now, most of them will produce these macrogametocytes because it only produces one. One merozoite will differentiate into one macrogametocyte. One will produce one microgametocyte, but these undergo gametogeny to produce, or undergoes replication to produce our microgametes. So we get our microgametes. And then release and penetrate, or release and penetrate. So now our microgamete gets released, infects our macrogamete, right, which produces our zygote. Inside that cell, inside the cell, you will then get cyst wall formation around this zygote, all right, producing our oocyst, and release from that cell. So now we have our oocyst inside the lumen of the gut. All right, and this is considered an unsporulated oocyst. It's this unsporulated oocyst that gets released in the feces. And then once we get out, and it encounters oxygen, then we undergo sporulation and sporogamy. Sporulation and sporogamy. So, our host is chickens. Couple things. Any questions so far? So it could have just traveled right down to the gut, right down to the cecum, get to where it needs to be, but that's not the life cycle. Life cycle penetrates, utilizes the macrophage, have the macrophage transport us down to the gland, to the livercule, where it then escape, penetrate, and start undergoing our morphology. All right, couple things. So we talked about sizes. Yeah, this is about 900. Then you get about your 200 to 300 merozoites, and you get even fewer for the third generation. Again, they're kind of getting bigger. The merozoites are getting bigger with each stage. All right. Our first generation, this release and re-effect spot takes about 2.5 to 3 days. All right, 2.5 to 3 days when we see our first round of morphology causing or rupturing and reinfecting our cells. All right, our second generation that gets released and reinfected is about 5 days. All right, so it's within a week. All right, we are now getting to that stage where we are getting to our merozoites that will undergo gametogeny. All right? It's at 6 days when our chicken can start shedding unsporulated oocysts, so within a week. Why is this important? Well, we're going to have cell death happen. What stages do we have cell death? There's one. Here's one. Here's one. Here's one. And there's one. All of those spots in our life cycle, we are causing cell death of that gut epithelial tissue. This is where we get major types of pathology. Now, I said this is self-limiting. It is, for the most part. It's programmed. We get to the third generation merozoites, now we will start undergoing gametogeny, but not all of them will. Some of them will reinfect the cells, and we have anywhere from 4 to 30 generations. 4 to 30 generations. However, here's where our catch comes into play. Most of our merozoites that get produced in these later stages get consumed by our macrophages and get taken out. So we do have an immune response that happens that helps to limit any of these. So really the pathology that we see in our chickens is coming from first, second, third microgametes, oocysts. Anything else is going to be just minor. So some of these you could have lingering infections, but in terms of those lingering infections, if that chicken survives to oocyst release, if it survives up to this point, then it typically survives the infection. It's just with these early stages, chicken's going to probably die in less than a week. Oh, geez. 5 days, 4 days, 5 days. All right. So what we'll do is we'll talk about pathology of it. I do have the image. This is an infected ceca. The infected ceca should not look reddish. That is lesions caused by Eimeria. All right, it's all, blood and stuff. It's pretty bad. Pretty bad. All right. Major life cycle review. Toxoplasma will actually break it up into two different life cycles. And we'll show the linkages. There, yeah. These aren't that great. See, here's my toxoplasma entire page. All right. I'll see some of you in lab. If you're reviewing this, please start reviewing this because our exam's next week. If you have questions, please bring questions to class because I'm sure other people might have similar questions. Have a good one.